how has it impacted my career? I think networking has been really important. I think I've grown a lot by meeting so many different and amazing women and role models. So I feel like I'm better positioned to grow in my career based on everything that I'm learning. So I'm very thankful for everyone that I've met through this community and others.
Women in Biotech, HBA, basically brought me to AAPS, and it got Ines into my life. And we just, we do what we think is going to reach our members, and we try to be thoughtful about, what would be most impactful for someone if they only have a limited amount of time to engage. So this year we're working to change our formats a little bit to make more pre-recorded. So like listen at your own type. So every time I meet somebody in WIPS, I'm like, wow, I've never heard of that role or that title yet, but here you are. So I think it's great.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin. I'm a life science professional from Kansas City. And along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. Chad is a bioanalytical scientific leader, and here at BioTalk Unzipped, we bring experts from different corners of the pharmaceutical, biotech, and medical device worlds to share their innovations, insights, career journeys, and personal stories to the show. Please do subscribe, and thank you for listening. Today is special because this is our first panel-style podcast, and today in the studio we have Dr. Ines Santos and Dr. Julia Sable, plus March is Women's History Month, and we all thought it would be a great idea to highlight women in pharmaceutical sciences and their allies. So without further ado, let's meet our panel. Dr. Ines Santos is a senior scientist in Translational Sciences and Precision Medicine Group at BMS in New Jersey, and vice chair of the AAPS Women in Pharmaceutical Sciences community. Ines received her PhD in biotechnology, analytical chemistry from the Portuguese Catholic University. Also with us is Julia Sable. She is an associate director at Kite Pharmaceuticals in the viral vector process design team, where she oversees the design of manufacturing processes and analytical testing for lentiviral vectors for CAR T therapies. She's also worked at other CAR T companies such as Team Unity Therapeutics and Legend Biotech, and worked with oncolytic viruses at Merck. Prior to industry, Julia was the director of virology for Columbia's ZMBBI Neuroscience Institute. She's based in Philadelphia and travels often for work. Julia is also the chair of AAPS Women in Pharmaceutical Sciences community. Welcome, Ines and Julia. How are you?
Great. Happy to be here.
Thank you.
Hey, thanks so much for joining us. So as we mentioned before we came on the air, it's our tradition at BioTalk Unzipped to make a $100 donation to the charity of your choice, and you both have decided on Steam Park. Tell us a little bit about Steam Park, what it is, and why it's important to you.
So Steam Park is a nonprofit organization that supports our next generation of scientists. So provides STEM support, like after-school programs for kids in the STEM areas. And I think it's quite important since we want to build our next generation and provide everybody with equal opportunities.
Yeah. Excellent. Well, I appreciate that. And we'll have a link in the show notes for anyone who wants to learn more about that organization and perhaps get involved. So both of you have had fascinating career trajectories. I'm curious, and I'll start with Julia, and we'll both answer this question. What initially drew you to science, and kind of what was there maybe a pivotal moment that maybe solidified your passion for this field?
I've been doing this a long time. So I think, most people, when they get asked those, say it was a formative teacher or something like that. I don't think that was it. I grew up in a fairly rural area in Northeast Ohio, and had a lot of access to, outdoors things. And, I think part of it is, at its heart comes from wanting to understand things, seeing, waking up in the morning in the winter and coming out and there's like deer carcasses. One of the things I was allowed to do was like help take them apart. And that probably was like the first, anatomy lesson that I had. And then for me, I really thought that I was going to go into medicine. That's what before my undergraduate, I wanted to be, a pediatric oncologist, which is definitely not, now not for me. It's a very tough field. And I was lucky. I ran into a mentor early during my freshman year. She saw me sitting out in the hallway. She was like, "Do you want to go play with some yeast and learn about genetics?" And I said, "Sure." And the first time an experiment worked, and you see your film, I was just so excited that I was like, I brought it home. I put it on the fridge, and I was like, I did that. Then I had to bring it back because it needed to be in my lab notebook. But yeah, it's just it's a high you almost chase because it's a piece of data that no one's ever seen before, and that's really that's the best drug, in my opinion.
I love those stories because we have a nice mix of listeners who are scientists and some non-scientists, and the non-scientists, I don't think they don't get it, right? And that's probably why they ended up in a different field. But I always think, wow, the scientists hear a story like that and they're like, yeah, the first time I saw my results from this experiment or that experiment, it just like grabbed me, and I had to have more. And that's not too dissimilar from maybe some of what I did. I'm an analytical chemist, but yeah, so that's really cool, actually. I like that.
Ines, how about you?
For me, I also don't think I had like a moment, or I never grew up like wanting to be a doctor or anything, but during my biology class in high school, I really love like learning new things. I'm always curious, want to answer questions. I also find it fascinating the way, the world is made. So I think my curiosity I knew I wanted to be in the lab. And then I think life just happened, ended up starting working in an analytical chemistry lab, and I fell in love with. I am one of those people that enjoys sitting at the computer and seeing a chromatographic peak come out, which maybe not a lot of people can understand. It sounds boring, but I get really excited when I see a peak come out.
Oh yeah. See, we're kindred spirits in the analytical chemistry, so I'm totally with you on that one.
Yeah. So that's how I got started.
I get excited and I also get sad, right? Because for me, for viruses, when you're washing your column, the size, the heightness, the area of your peak start to make you really nervous at some point. So
that's awesome.
Yeah. No, thanks for sharing that. That's it makes a lot of sense. So what's maybe one project or discovery you've worked on that excites you the most or excited you the most?
That's a tough one. A project that I worked on that excited me the most. I feel like any project I worked on, I'm very excited about.
How'd you pick your dissertation research? I mean, hopefully that was exciting for you.
And I kind of also wanted to bring about like the way I came into the U.S. and also I'm very thankful for Dr. Kevin Shook. Like I did an internship during my PhD in his lab and working on mass spectrometry. So I had.
And where was that?
UT Arlington. So I had no mass spectrometry expertise or knowledge before that. I had a friend, and at the time she told me, if you want to get a job, you should learn about mass spectrometry. So I thought, okay, then I'm going to go do this internship in his lab and work with mass spectrometry. And I'm thankful that he accepted me in his lab. He could have said no. And so I think all the projects that I did in his lab, one of that stands out was a collaboration with actually LabCorp, working with Dr. Russell Grant, and it was not mass spectrometry related. We were, at the time, were working with JCVUV, analyzing fatty acids, but I love that project. The collaboration with the industry was my first collaboration project. They gave me an idea of what working in industry is like, and also that we were working also to answer questions that are important and helping patients. So I think that project that stand out for me, and also very grateful for that opportunity. Yeah.
And Russ was a guest of the show about a year ago. He's a good friend of mine, and anybody who listened to the episode, and certainly, Gregory getting to know him when we did that, I bet, we can attest it. I bet he asked you some tough questions that made you think hard, right? Like,
right.
yeah, really test your brain. I mean, so thinking about, that's awesome. That's awesome. Yep.
Excellent. Julie, how about you?
Oh, I've been lucky. I have many interdisciplinary things. I guess if, from my perspective, when I was the director for my own lab at Columbia, I got to work with amazing neuroscientists all the time. They would bring me their problems and say, I would like to be able to track, the subpopulation of neurons. I've got a really complex idea. I need, 5 different color fluorescents plus, some other bells and whistles on it for tracing. So anybody who walked through my door always probably had what would be the start of an exciting project. So, for me, it would be from the initial cloning design, from the molecular space to manufacturing the virus for them. Doing a lot of this requires multiple viruses. It's always fun to tell people you make rabies as your side job, as your day job. It's a great monosynaptic tracer. I would design a lot of things that, I would ultimately get to have multiple groups answer very different questions with. So for me, that was certainly fun. I've also had the opportunity to run the Center for Hep C at Rockefeller University for Charlie Rice. And I have to say, it was really lovely to see him win the Nobel Prize for the group's work historically on the discovery and the models basically for understanding Hep C replication. And now that I'm in the pharma space in particular, I think a lot of my current role, my current company, Gilead, although I didn't have a direct connection to it, watching them do lenacapavir this year was amazing. That's one of the coolest things I've seen in terms of virology. I'm a virologist. So
Sure.
Looking at a really different target for a long period of time, those guys kind of figured out how to go after the capsid and make a much better product that has, long-standing efficacy. And I think that's going to make a huge impact for HIV over time. So lots of different good ways of thinking about projects.
Julie, was lenacapavir, is that a small molecule drug, or is that it is,
but it's a small molecule,
but it targets the HIV capsule?
Yeah. Capsid.
Yeah. Okay.
It's an envelope virus, and it's got kind of like a hard candy inside shell.
Okay. And do you see that becoming a new standard of care for HIV, or?
I think it's going to stop the reservoir. So 2 things they did with that study that were really powerful, especially in the space that they did in Africa. Women and pregnant women in particular are infected at a very high rate, and they were able to do their study design specifically with that as their core cohort. And it overcomes a lot of serious social barriers as well, going twice a year for a shot as opposed to daily medicines. Not discounting those, they all work. But I think that if you really want to, end HIV or you really put a solid dent on the reservoir, you have to find a way that is socially acceptable and also, long acting.
That's great. It's a incredible treatment.
Yeah, for sure.
In your role in project management and associate director for virovectoral process design at Kite, you've been involved in addressing some CMC challenges in cell and gene modified therapies. What are some of the most pressing CMC challenges you've encountered, and what are some strategies that have proven effective in overcoming those?
So many. I wish there were less. You're making a large, well, I call the viruses like large proteins, right? And from a modality standpoint, some viruses are easier than others. Some of them have envelopes, some of them don't. For CAR T in particular, that we use lentiviruses because they integrate. And when you're processing that, you have to design a process that's gentle and fast. And those are not necessarily easy things to do when you have to pump fluids over, through small small orifices. You have to think about a gentle way to capture and remove impurities while not damaging your virus. There's each one of the upstream steps has some aspects when you're in the reactor, and then the downstream is basically a series of shear stress that you try to mitigate all the way through. And then when you get to the end, it's about how fast you can fill finish before you start having biological activity drop off. So from my perspective, any virus fits into this, but lentis are particularly painful because, there are envelope proteins on the outside. They can be damaged and degraded over time. They can be masked by having high impurity loads. So the main ones are, looking at any process that you're designing to try to keep your impurities as low as possible. And also, try to have the least amount of processing steps, if at all possible, to reduce the total time. That's my advice for anybody who's coming into CMC. You as fast as it has to be and as gentle as it can be is the motto.
Julie, have you been in manufacturing for the majority of your career, or for a good chunk? I mean, I'm curious how you ended up in the CMC side.
I didn't know much about. So I had a partner who took a job at J&J, and I applied to one job at Merck, and it said vaccine process design. I had never done CMC. I had no background in it. I mean, I'd been making viruses for 20 some years at that point, but I'd never made them at that scale or at that with that level of this. Oh my goodness, this is going to go in a human. So everything I was making was going to go into a mouse or a bird or something like that. So it was an education. It was the best education. So sometimes you make a mistake. I would say that for me, I landed in the middle of a brand new oncolytic virus program that they had never done. These are all people who make vaccines, live attenuated vaccines, and here's me with a coronavirus, basically ready to wreak havoc. So I had to learn everything from safety, for my team and myself for handling it, to how GMP works for cells and seeds, and how the whole drug product, final form finish works. I thought I knew a lot about freezers and, putting things away and making them stable over time. I knew very little at the beginning. I learned a lot very quickly. And I have to say, if you're looking for a formative way and you're not sure, a lot of people I see try to go from their postdocs into something that still looks like a postdoc position. They're like, I want to go into discovery. I'm like, please go into CMC. Your creative problem solving, especially women, there are not enough women. Bring your creative problem solving to something that looks like engineering, but it's basically virology and cell biology and biophysics sitting underneath all of it, because your problems are always embedded in that. So for me, whenever I get the opportunity, like now, I put a plug in to say that CMC is like a great space to be in. You can also stay in analytics and be involved in CMC, because you need assays for everything that you're doing. How are you doing? The analytical portion of it needs to be there, lock in step with what you're trying to achieve. So I'm in it now
advice.
7 years.
7 years. Okay. So relatively new. And I think
I'm a baby in CMC land, but
okay. From my perspective, only 7 years sounds like you're pretty new into the space, but it seems like you've fallen into it pretty nicely. Yeah.
And Ines, in your recent publication on the application of a supercritical fluid chromatography tandem mass spec for chiral bioanalysis, you demonstrated the robustness and reliability there. What were some of the key challenges that you faced during that study, and how SFC MSMS compares to maybe traditional liquid chromatography and chiral separations?
Yeah, I think the main challenge is just to prove to people that SFC, it's a robust technique. It's not what it used to be. Honestly, I'm doing the work. SFC works quite well. So there's not really like technical challenges with it. I was really glad I could, apply this technique, and at least for this field, it's a novel thing. It's used in not any other field like purification. It's very used, but not in bioanalysis. So I thought I like take on the challenge to show that why aren't we using SFC in bioanalysis as well, when SFC can be faster in certain applications like chiral chromatography can be greener. And if we're looking for high throughput, like why aren't we using it, right? So I really enjoyed it, and I enjoy showing it to people and trying to help spread the use of this technique in this field and also in the validated or regulated bioanalysis.
So answer that question for me. Why aren't people using it more? Because I actually had a publication maybe 2005, 2007, something like that. I don't even remember when, but and I thought at that time, like, hey, this is going to start getting used more. And then it was, and it didn't. So why should it get used more? And do you think it will?
What I hear is that there was like a spike in SFC many years ago, and then it went down, and I think now we're back up again. So I think it's up to the newer scientists to honestly adopt the technique and start using it as an LC. And I'm hoping I can help with that. I think people still think of SFC as, I don't know, at the time when you used it, if you faced challenges with the technique, but there used to be challenges. And I think people think that you have to be skilled, you have to be knowledge, and sure, you have to know what you're doing, but it's not, in my opinion, it works just like an HPLC. So I think people just need to start acquiring the technique, using it, feeling comfortable with it, and just go with it. Hopefully we keep using it.
Yeah. It's interesting that it hasn't grown more, because it was mostly practical challenges, right? Of just you're dealing with a supercritical fluid, right? Which is almost more like a gas than a liquid. And so, it just leaks and making a good seal, and you've got a tank of CO2 instead of a bottle of liquid. So how do you tell when it's empty? And it was like all these practical challenges. And then I think it's a little bit damaged by the perception that it's only for chiral analysis. And I think the green, the opportunity for it to be a green technique is actually a huge opportunity as well, as we look for more and more opportunities to be a little bit more environmentally friendly. It's sure a heck of a lot better than the gallons and gallons of acetonitrile the HPLCs
and I was just
going to
say that the SFC is not just for chiral, right? There's a recent publication for oligonucleotides. Also, I'm working with biomarkers with that technique as well. So we're showing that, yes, if you acquire an SFC, you're not stuck to doing chiral bioanalysis with it. You can apply to many other applications.
Yeah, excellent.
Very good. Well, Chad, it's that time for new news.
New news. All right.
Very good. In Fierce Pharma, on February 12th, Pfizer's blockbuster ADC Adcetris nabs the FDA approval to treat large B-cell lymphoma. And this was, again, Adcetris secured their approval for treating LBCL in patients who have undergone at least 2 prior lines of therapy and are ineligible for CAR T or stem cell transplants. And this approval based on the phase 3 Echelon 3 study, where adding Adcetris to lenalidomide and rituximab reduced the risk of death by 37% and extended life for a median of survival of 5.3 additional months compared to the standard treatment. And the combo also showed a 47% reduction in disease progression or death, which is great. Again, this article mentions that patients must have tried at least 2 prior lines of therapy and be ineligible for CAR T or stem cell transplants. Why is that important to the success of this new ADC, do you think? Is it so they don't compete with existing CAR T therapies, or something else, or both?
And it's not it's not a quiz.
No, it's not a quiz. Just kind of curious what your opinion is on that. What do you think?
And I was kind of curious as well. I know, the companies you're in are, obviously, Julia, you have a lot of experience with CAR Ts, and Ines, BMS does a lot of ADCs. We're not looking for sort of your company line on that, but I was really curious also with looking at this and reading about it, because thinking about all the new treatment options that are coming out there, and then when you look at, with clinical trials and how do they go down that path and that decision path. So just kind of curious what you guys think about that, more so than it's a quiz show, because I I saw the look on
Totally.
both of your face, like, oh,
Uh-oh.
No, I'm not a medical doctor, and
yeah,
I'm not
no, we're not
speaking
looking
on
for that. Yeah,
behalf of
Right.
Kite, but I mean, obviously, the company I work for is, has treated more than 25,000 patients for CAR T. So, there's different types of LBCLs, and there's different pathways to it. So, as CAR Ts have progressed into earlier lines of treatment options, there are still, for some people, it's a permanent, cure, and for some people it's buying you time, right? So the refractory aspect is something I would say the clinical field, I'm not a medical doctor,
right.
doesn't
Mhm.
understand very well. Like
Mhm.
when we bring patients in, we're trying to take as much information as we can and try to do as many diagnostic things as we can, but we don't always chase our patients for a really long period of time. We do what we have to do, but like there's not the opportunity to keep pulling people back, and we don't have animal models that live long enough to really kind of model the real refractory state. So when we're making new CAR Ts in particular, right, that's the that's the population basically. So it's what is your binder specificity, and I would say the ADC this is where I would compare ADCs or T cell engagers or any of that things in the field. They're going to have different approaches, right? A T cell engager is a big one, and the ADCs right now, I'm not a small molecule person, so I don't always, or biologics person, so I don't always know exactly what it looks like structurally. But my understanding is, when it's in the body, the type of dose, the patients and medical doctors try to make different decisions. CAR T is, as it takes time. You got to get person's material out,
Yes.
process it, get it back to them. So there's maybe the appeal that, if there's some efficacy in buying more time, a single shot might be more attractive. If you're arguing for that, then, in vivo CARs, which are coming right now, they're in active clinic. Those are really going to be highly competitive to it. So I think that it's good that this space has so many options. They all have some good pros and cons, right, to them, and it's up to the patient and the doctor to decide which one is right for them. Within the CAR T space, like we're only seeing like 20% of the patients that could benefit from a CAR T are taking it. So that means the other 80% are taking things like these current modalities, maybe with the hope that, they have long lasting effects. And a lot of time it just buys time, is my understanding.
Yeah.
So it's not that one is better or worse. It's just each person, probably has their own output, outcome from it. And for some people, like I said, it's amazing to hear a person who gets CAR T and they're like, I'm cured. I want the same thing for a person who gets a biologic modality or, an ADC or a T cell engager or a BiTE, and they get the same result. That's what we all want. So from my perspective, we don't understand the biomarker refractory space really well. We're trying. I think all these companies, larger pharma and really small biotechs, are trying, and they're designing better targeting mechanisms to try to hit the whole population as fast as you can.
What's amazing when I think about these two kinds of treatments, the CAR T as well as the ADCs, is 20 years ago, which was really not that long ago. ADCs were in the very, very early stages of development in pharma, and CAR T was a dream in somebody's research lab in a couple of universities probably, and to now have both of these therapies available, both of these types of modalities available to the physician in their war chest, which one of course, like the CAR T is essentially a cure, right? We hope that it's a cure, and of course the antibody drug conjugates, that's for the non-science listeners, that's the missile delivering a payload to blow up cancer, right, and just knock it out of the park. So just think about 20 years ago, this wasn't even an option at all. Is amazing. And I don't know if you have some other thoughts as well. We don't want to.
Good. I mean, I think Julia spoke very well about it. I agree with you. This is amazing what we're doing nowadays. Yeah.
Yeah.
I think so for heme malignancies, this is we know that this works. I would like to see or think I don't know much about this, but I do know this tumor microenvironment and solid tumor microenvironment in this instead of heme, right? There might be benefits of having both, right? Just because you have CAR T doesn't mean you can't have another thing. I think the best combos might come from having oncolytic viruses as a palate cleanser, if you will, or an activator, and then chase with a CAR behind it. And I would say you could do the same thing. Like you need to have access to these tumors in the solid environment that will, that have, it's like a jawbreaker, whatever, those candy layers with all the layers.
Yeah.
That's really what solid tumors are, right? So you're going to need, I would like to see more combination studies where CAR T is not just the variable, but I think that's a far way off right now, unfortunately. And a lot of times when you get a patient, I understand when they come into a study, they might just be coming off of one, and then you give them another one, and you're trying to sit there and say, is this because of the thing that they're coming off of, reduce their tumor burden, or is it my new thing I just put in them that's doing such a good job of further, reducing it? So people come to you, come into their treatment at very different spots. So I think for that particular drug, it's appropriate,
Amazing.
That makes sense. Yeah, it sounds like it's a little bit more of a last resort after, the CAR T and maybe other stem cell opportunities have failed. I just thought it was kind of interesting when I first read it. It's like, well, they have to have tried something else first. But I think that's really the optimal approach. And then you bring the ADC
a lot of times,
extend life.
Yeah, the refractory patients from CAR, right? They lose their antigen, right? So our tool chest needs to get a lot more specific for targeting. And that's the other reason I thought in vivo was kind of interesting, right? Because instead of using, a CAR-based, the CAR on the outside of the patient's T cells, now you just put that on your virus and you can make it, CD, whatever. You can make it, you can put a targeting molecule on the outside of your virus itself, that instead of having to depend on having your CAR on the outside of a T cell. And the younger the T cell, the better the therapy. So,
Interesting. Well, let's shift over to AAPS. You're both leaders in the Women in Pharmaceutical Sciences group of AAPS. And Ines, I love your T-shirt, by the way, Empowered Women. That's awesome. What? Oh, yeah. Empowered Women, Empower Women. Very good. Excellent. I didn't see the second half. What inspired you both to join and kind of lead this group, and how has it maybe impacted your career? Ines, I'll start with you.
So this is a theme that it really speaks to me. As a woman in the field, and I want to make sure we all have the same opportunities, sometimes you go through certain experiences and it's good to connect with other women and see, if does everyone go through the same experiences, ways of brainstorming, how can we approach certain things and how can we make things better for all of us. So I wanted to join this group to be able to, connect with other women in the field, learn from them, and also be able to help and make sure we provide women in the field with the same opportunities to grow in their careers. So that is why I joined. And how has it impacted my career? I think networking has been really important. I think I've grown a lot by meeting so many different and amazing women and role models. So I feel like I'm better positioned to grow in my career based on everything that I'm learning. So I'm very thankful for everyone that I've met through this community and others.
Excellent. Julie, what got you into leading this group?
I got there peripherally from other women in science groups. So I started in Women in Biotech, which has a strong presence in Philadelphia, where I'm based. And I did some, mentoring circles and some other things. And I was actually, actively posting things on LinkedIn to help people like me who just didn't understand roles and levels. And the woman, Viva Jawa, who was our what is she now? Our past chair. She reached out to me because she was reading them and she said, this is really interesting. Would you be interested in speaking about that at an AAPS, panel discussion. And so it was my first time engaging with AAPS. I wasn't a member at the time. And then last year, I became vice chair, and I got to, work with Ines, who has just been phenomenal in bringing in really interesting ideas and panel discussions throughout the year. They, the topics, and particularly allyship for this year, is something that we're really excited and focused on. We're not here just, we don't work in a vacuum. It would be kind of dope to have an all-lady lab all the time, but that's not the case. I wish we could, but we don't. So we end up having to work within the structures where we're at. So that's the central theme, I think, for this year that she brought. As again, we're not recycling, but we're inspired by topics from other places. So HBA, for people who are listening, I think HBA is a great community as well. Those are healthcare business associates. I don't even remember, but but women in biotech, HBA basically brought me to AAPS, and it got Ines into my life. And we just, we do what we think is going to reach our members, and we try to be thoughtful about, what would be most impactful for someone if they only have a limited amount of time to engage. So this year we're working to change our formats a little bit to make more pre-recorded. So like listen at your own type, not necessarily podcasts. We don't really know podcasts like you guys do, but similar kind of idea. I think you get inspiration from, for me in particular, I listen to a lot of podcasts and take inspiration from them. So we thought about how we could modify our format. And our members in AAPS are across, the United States and probably global as well. So you really can't do a lot of in-person things. We do have once a year at the PharmSci 360, and we get to meet in person with a lot of the people that are part of our Women in Pharmaceutical Sciences. And it's a great opportunity to, to reach, a diverse group of people. I couldn't say thematically. I don't actually know from our membership. Maybe Ines knows what our breakdown is like for disciplines, but it's broad. So every time I meet somebody in WIPS, I'm like, wow, I've never heard of that role or that title yet, but here you are. So I think it's great.
But that's fantastic. So, I think one of the areas that I see a lot of women, really focused on, which is fantastic, is the mentoring and the development. And Ines, you mentioned that. So I'm curious both about, your role in the group and how that works with especially the younger women in the group. But also there's hopefully some people listening to this are like, oh, I'd love to get involved with that. How do I break into this group, right? Some people are kind of daunted by something that I think you're going to tell us is a pretty easy process.
Yeah. So you just have to be a member of AAPS. And then as a member of AAPS, you have multiple things you can receive, but one of them is be part of different communities. There's the bioanalytical community, and then there's the women in pharmaceutical sciences community. So you just choose the community, and then you can be part. There's a, we can post on the website. If you're interested in joining, please add me to the meetings, and we can just add you to the meeting. So very easy process to join. And in terms of mentoring, I know AAPS provides a mentoring program as well that you can register and be involved, but also coming to our meetings. You can always reach out to us or anyone in the meeting. And if you want to have one-on-ones and say, I want to learn more about what you do, or I need advice, or I just want to, chat with you, people are always willing. Like I always hear, people love to talk about themselves. So always willing to talk to you. So there's that way you can reach out for mentors.
That's fantastic. And how, Julia, have you had an opportunity within this group to help mentor? I mean, do you see a lot of younger women coming into the WIPS group?
So Ines did a really good thing. She sent out a survey to all of our current membership, and it was really informative. So if you're running, a sub-community like this, I highly recommend polling them intermittently just to see what it is they want. And for us, we do get asked for mentorship. We, like we said, AAPS has a formal mentoring program, so we defer people to that program. we have what we've noticed is we have sort of subpopulations, right? People who are postdocs who, are in AAPS and are thinking about, applying into, looking for pharmaceutical roles. We have graduate students as well. So what we're encouraging our membership to do this year is to self-assemble. We have no rules about this. We want them to meet as much as they feel like they need to. And having, as much as simple as it could sound, like a WhatsApp group chat when you have questions. It's about forming, sub-communities within our larger community. People are at a lot of different levels as they come into this group. Some are mid-level, some are, exiting. And those people tend to circle around each other. And I think that we actively encourage that. And then we try to make sure that, like we said, when we have panels or discussions or invited speakers, that we're mixing it up so that across the year, there's a little bit for everyone, but we actively tell those subgroups, like, tell us what you want. If there's a person that you think has really spoken to you and there's a way to get them in, that's one way. Or you can just be sharing things and resources, books, podcasts, TED Talks, things like that, like YouTube's that you've, feel free to post it in and share it. We have AAPS has a unique setup where there is a way that you can upload, pieces of information and links to things. So I think that
through the community pages, you can upload files and information and then certainly the community chats and things like that. I think that's something where I've thought, being part of some other communities, I thought AAPS has made a really good forum. And it's one of the things I think in general with AAPS is really, you need to just be motivated and have an idea and want to share and want to work together. It's always, I've really respected that it's really an organization for the members to get out of it what you want, and like so many things, you get out of it what you put into it. So I imagine the WIPS group is similar. It sounds like it is.
Yeah, I would say that our engagement through like the portal itself is a little bit less so than say like individual communities. So if you're new to AAPS, you're going to see a lot of activity in certain groups. I'm in the cell and gene therapy, for example. So, those are your phone of friends. Those are discussions you would follow and, maybe not engage with, but follow, read, look at. I think what we are, for me, my personal aim is to put things in that will, even if they don't speak to it in a chat, they leave with a thought from it, or they take something. So once a week right now, we're doing using a social media platform. We're using LinkedIn, and we tag AAPS WIPS for anything that pops up. So for me, it's part of my own development thing. I look for things that I found interesting or inspiring. I summarize them and put them in, but I also encourage and solicit from my own team. Like, did you like something? You can do it this week. So or this for like this month. And that might be what we end up doing later in the year is actually taking some of these subgroups of people at different times and just handing the mic over, for lack of a better word, and saying, you've got a month, pick, once a week, pick something that you did. And a lot of times, at least for me, it's hard to make time to do that, but it's it's a commitment device to me to say, it's important for me to be growing, learning, thinking about new ways of doing, executive management or, mentoring. So whatever it is, sometimes it's very sciencey, sometimes it's less so.
Yeah.
But that's what we're trying to do is, it's a real community. So you need to listen to your community and be open to what your community wants to talk about.
Right. And I think
I don't see a lot of people engaging with it, but they read it. And they definitely see it, but I don't get a lot of comments on it. And it's not that I feel bad about it, and that's not my goal. Like I said, it's my commitment device to me. And I think putting that through is something that, I just think that it's a way that anybody could do that for themselves. And they also helps you engage with your scientific community, which is, through LinkedIn, it's a lot more focused to the types of people who are in your own immediate sphere.
Right. And I think it's really important what you said earlier about these subgroups and trying to build some engagement? Because that's, I think, one of the hardest parts of any group, is you come in with a great idea and people agree with you and they join, but then getting them to actually engaged and talk to each other sometimes is the challenge. And I like the WhatsApp idea, because then it's open, it's on your phone, it's open to a global audience, and I've seen some success in other subgroups happening with that. So that's really good.
And can I just mention, when we talk about mentoring, we want to be inclusive, and like it's not just women mentoring each other, but also, like you've said, allyship, having men mentoring as well, or being mentored, right? I'm very thankful for a lot of men I have in my life and professional life that are my mentors. So also want to highlight the importance of that.
Yeah, we appreciate that. That was one of the topics Gregory and I had talked about is how can men be better allies? Because as someone, I know that I try to be sort of sensitive to thinking about inclusiveness in many different areas. Today we're talking about women, but what are some things maybe where you've seen where, like, how, maybe this guy could have done a better job, or maybe it's something that maybe it's something Gregory and I could have done better in planning the podcast. I don't know, but we, at least I should only speak for myself on this subject. Certainly I always want that feedback, but I don't think I always get it. And I tend to be somebody I just like plow ahead. So I don't always notice it. So
I would say, like, it applies to everybody, right? Not just for women, but for example, I think if you're in a meeting and someone speaks over someone, or if someone gives an idea and then the next person gives the same idea, but only that person gets the credit, to try to speak up, right? And like I said, it applies to anyone. I feel like anyone has experienced this, right? To not be silenced, like, oh, it wasn't with me, so why should I bother? But if you can speak up for others, that means a lot and helps, make the world a better place.
Yeah, I love that. I love that thought, because that's just encouraging, I'll say, diversity of thought and experience and inclusiveness just across the board, right? And if you do that across the board, then you're I'll say you're covered for women, sort of, for the most part. But if you are just considering everybody, do you think you guys in your education and growth and going through a scientific development, did you experience special challenges, do you think, as women, to be as successful as you have been?
I think the
answer's always yes.
Go ahead.
Go ahead, Ines. No, go ahead.
No, I mean, I just think more like sometimes people like to put you in boxes, right? And I think for me, that is a little bit of a challenge, to trying to get out of the box of, people see you and they labeled you, this is what you're good at. And I think that's one of my challenges, is to break that barrier of, I want to do more, and this is what I want to, I don't want to be in a box. I want to be in a circle or whatever. But I would say that's one of the challenges I feel like I've faced, to try to prove myself in other areas that maybe people think women aren't so good at.
Well, what I've learned from both of you guys in this discussion so far is, I don't think either of you want any kind of boundaries, circles or boxes or anything, right? You just want to, knock down all the boundaries and take advantage of all the opportunities out there. So,
Go ahead, Julia.
I mean, I think, Ines is right. Like, if you're amplifying, if you see somebody, taking credit for someone else's idea, it's a common one that happens, unfortunately, still all the time. Speaking to it, just, being direct and saying it out loud, because everybody who's leaving the room knows that, maybe it was suggested at first, but then it was like the person who's taking credit for it and walking out the door with it is just, basically parroting something they just heard. I like amplification. I think, to your point about allyship, if you hear a good idea, you give credit to that person first and say, I really like that idea, instead of just repeating what that person said. So I like this because, right? So give it more support with a specific example and a justification why it is, you're saying yes to someone else's idea. It's not enough to just, repeat that person. You have to acknowledge their contribution. And if you have something else to add, great. Then you're problem solving, providing solutions in a complementary manner, not as a competition. And I think a lot of times still sometimes feels like a competition. In my role in CMC in particular, there's not a lot of women at the executive level in particular. So it's often being the only woman in the Zoom chat or being the only woman at the table. A lot of times, I find myself listening and moderating and, trying to keep us out of rabbit holes and things like that. And that can be allyship too. I don't have to be the only woman in the room monitoring the time. Our time is very valuable when we're in these meetings. And, if somebody is really consistently going off on a tangent, it doesn't necessarily have to come from me. Like if you are a male in the room and you notice your colleague is consistently going this way on something, you can have that talk with them too, because it's a lot more challenging for the woman to go to a man and be like, listen, you're constantly just like derailing our agendas or our things like that. And it happens, there's that feedback mechanism should not have to be centered, on the female who's in the room. Watching the emotions of a meeting can be something that, again, a woman can be expected to modulate it over time and bring you back and kind of keep you level. Guys can do that too. Good leaders do that. I'm not saying that this is, something systematic or even within my current workplace. I'm speaking about the 28 years I've been working in science. There's all kinds of flavors when you're in these meetings, when you're interacting scientifically or having discussions. You can have proactive, disagreements and still be civil to one another and not bring it to gender, right? It can just be, I see this person. I think maybe that's for me the most important part of allyship. It's not that she's a woman scientist. She's a scientist.
Yeah.
And I respect her the same way that I would respect any other scientist. And that's all I want. That's all any of us, I think, really want, is we want to have that equality inside the space. It's not that I don't want to, be a woman. I'm a wonderful woman. I like being a woman scientist, but I really want to be judged based on the merit of my work and the quality of my science. And I want to be seen for that. I don't necessarily need to be seen, like you said, in a box or a bubble or a heart or whatever.
That really, so much of what you guys both said has really resonated with me. And I was talking a little bit before the podcast with Ines about a couple of different things. And one of them was that, I learned so much from these discussions every time I had it. I was trying to think, what am I going to take away from this discussion? And what I'm really taking away right here, because I try to keep it simple, is in that effort to be a better ally. It's not just me changing sort of like, do I need to treat women better or something like that? I think generally I'm pretty good, but I think also supporting you guys when somebody else, isn't overtly, being, sexist or whatever it is, misogynist, but it's when you're just being overlooked or maybe I was trying to think of examples. Maybe the lady on the team is the one who always is asked to take notes, right? Like, why? I can imagine that would be a thing that would happen that the guys in the room would never notice, because it seems normal. And so trying to recognize and be cognizant of those things, both with the teams I'm leading, as well as the interaction of what's kind of going on around the team. So so again, thinking about what can my takeaway? How can I learn from this discussion? That's one of the big ones for me.
Yeah.
The good news is now you can use an AI chatbot and nobody has to actually take minutes,
Right.
It's maybe a good, this might be like a thing of the past in 2 more years that people will, it won't be, to one gender role or another. And the other part of it, I think I just want to add is, when you're in the room and you're watching your team and you don't see, you have a subgroup that's just not engaging, not speaking up, never ask questions. I was a university professor for a long time as well, at Columbia and Barnard and Cooper Union. And I had classes of 120, I had classes of 10. There are different ways to get people to speak up and engage. And I think that there's a thing to be learned from the academic space about, or social science, maybe in general, org dev, like how do you, what are better ways to solicit feedback and ideas? And one of my favorite ones this year actually came from Adam Grant, a podcast I heard from Adam Grant, but it was instead of putting it in a group setting and what do you call it? Like, bringing everybody together to try to group think a solution to something. They suggested having each person individually type something in or come to you one-to-one with their ideas. And you got much more creative problem solving across the board, because if you make it a requirement, like everybody has to give me some feedback on this, that it actually caused a better outcome and less traditional kind of, solutions. So I thought that's a great way for allyship to work as well. Instead of putting everybody in a room or a Zoom chat and expecting everyone to contribute equally, find alternative ways to bring in, the ideas that you need. And sometimes, like I said, it can be one-on-one, it can be through a quick email or a Teams chat. You can do, polls in Teams that are private. Love those, use those a lot. Whiteboards in Teams, same thing, where they write it on a Post-it, but they don't put their name on it. So I don't know who wrote it. I do that a lot now. I use a whiteboard and Teams with a Post-it approach, and I can't tell who put the idea up, but I can at least say, okay, these ideas are over here, these ideas are over here. And ultimately, I'd love to know who came up with it, but I thought that was kind of, it's almost like with resumes. They say, don't put your, identifiable information on it.
Yeah.
Okay.
Right. That's interesting, because I'm thinking, both in meetings, like you said, but also in the scientific meetings that we go to, the format, and, 99% of the cases I've been to is, all right, we've got a room of 100, 200, 500 people. We've got someone speaking. And then, hey, you have a question, go up, stand at the mic and talk to everybody. I think that eliminates about 90% of the scientists in the room who just won't ever do that. But I would also guess about 90% of the scientists in the room have questions, right? And so, if we could think about ways to be more engaging in that way, allow everyone to get their voice heard, and that would be so much more inclusive of an approach. I think that's something we need to think about, and maybe we can take it back to AAPS to try and figure something out for some of those meetings, because I'd love to get broader feedback. Yeah,
I've seen some technology where they do allow you to type in a question, and then there's like someone who is facilitating that and trying to get those
moderating.
Yeah, exactly.
Yeah,
and get those to the speaker, so that helps a little.
Yeah, if you're the speaker, you'd want to see all the questions that popped up, right? Because it can help you think about your data and, and really the next best idea. Because I think beyond in the room, the breakout sessions and the other things that they set up, again, you have to have a level of extrovert into you that will allow you to go up and say something. And, I have it, but I even know, like, I'm respectful of that person's time if they've already got 6 people standing around them trying to have a conversation. Often what I've done, and I think I continue to do, is I just type it in an email after the fact. Hey, I heard you talk. This was really great. I thought about this. Do you know these papers? Blah, blah, blah. That's another way to network too. So, but that still requires an entry barrier to get to that person, find their email, all the other things. But I really like that, in education, we call it an exit ticket. So before you leave the room, as part of doing attendance, you get you have to write a question down about something you didn't understand for the day. And I almost want to do that for a lot of meetings, especially like scientific content meetings, just to debrief and say, if this was unclear, I'm still not clear about X. And I need everybody to be clear about it, not for taking a test in this case, but just to have clarity on a subject matter, a technical matter, or even a policy, right? That, we're putting through. Have it be, give them an exit ticket, see what happens. See
I
what's
think it's
still
a great idea.
undone.
Yeah.
Yeah.
A great way to marry the educational techniques into, applicable scientific business, right?
Mhm.
I have to
It's
say my college students really hated it.
Probably.
But I would
But-
use it. You can always use it to open your next. So what you do with your exit ticket, you figure out what the gaps are. You can go back and repeat things that need to be repeated, and then you can also, individually find people and say, "Hey, I see this question here. Let's talk." Right. So it can give you a connectivity too.
Well,
And
Julia, we aren't quite there yet, and I'm not going to ruin the surprise, but we're going to give you guys both an exit ticket as well. But
Okay.
but I don't think we're quite there yet.
Great.
Almost. I did want to make one comment about when people are taking advantage of a situation or maybe somebody's idea, and everybody knows it was someone else's idea, but they didn't say anything. It's one thing. There's bad in the world, people doing bad things, bad behaviors, right? But what's worse, the people doing the bad behavior or the good people who stay silent and do nothing, right? And that's kind of what resonated me through that. So if there's if you see something, you hear something that's not right, say something. It doesn't have to be a huge confrontation. It's just there's always approaches to make it professional, polite, and still get your point across, right?
That's right.
Excellent.
I was just going to say one thing. I don't know. Just back at being a colleague or a manager, I think also don't make assumptions, because a lot, for example, women that just had a baby, sometimes people make assumptions like, I won't give you this project because you're probably too busy, like right now you probably don't have the headspace. So I would say don't make those assumptions. Just speak to the person first and ask, like, do you have the bandwidth? Do you want it? Provide the support if the person has a lot going on, but they do want to do things. Women still want to develop their careers, even if they're busy at home, right? So I would just ask to, regardless what the subject matter is, don't make assumptions, to always speak to the person first and ask what they want.
That's a great point. Yeah, thanks.
And I think there's allyship at home too, right? Like if I've agreed to something, and I know that it's important for me, for my career, for, science. I've got a teenager and I've got a partner I live with and 2 cats, and they all want to see me too. So I try to, from my side, my responsibility, I try to say, I've said yes to this. This is what I'm committed to. I, promise to try to keep within these boundaries. The next time, sometimes like RTQs will come in, right, from a regulatory agency, and you have 48 hours. There's nothing you can do about that. So when that happens, my obligation is to tell the people on the other side, including the people I work with, but especially the ones at home, like, this is happening right now. This is short. It's going to be, I'm going to look a little stressed, as it might be a little unpleasant because I'll be working a lot. But when it's done, go back to reset. And I think that's something I'm learning to do very late in life. That's helpful for, the reciprocity part of it, is that sometimes science is not a 9-to-5 job. Sometimes science happens globally in my company. I think that setting those expectations helps with, and then also having good boundaries on my side to say, I can do this up to here, but I'm shutting off at this time. And that's another thing, for allyship that you can kind of always respect. And I work for a company that's wonderful. I have a UK side, I've got an LA side. So in Philly, I'm getting it throughout the whole of the day.
Yeah.
But most people acknowledge where you are, and they are very respectful in asking, and try not to make it abusive. So those are all things you can do on your day-to-day to make it easier, right? To Ines's point, you don't know what that person's situation is,
Right.
and what their obligations are when they need to be, on or off at a given time. I don't really have a lot of questions. If somebody says I need 2 hours off, I'm like, I don't even know what it is for. Go ahead. See you later. It'll get done.
Need it. Yeah. You're a professional.
Yeah. You never know what someone's going through.
I think that's a big thing that's happening this year that maybe you guys will speak to later in other podcasts, but the back to office, especially for my colleagues who are in LA, it's bonkers. it's 2.5 hours to come in, 20 or 30 miles sometimes.
Right.
Yeah,
Right.
it's the traffic is worse somehow than it was before the pandemic, and it's so for some people it's really making it very, very difficult, without a doubt. Yeah.
Yeah.
To me, that's allyship as well, right? Like
Mhm.
if you're,
Mhm.
and it can
Yep.
be everything. You can have
Yep.
pets, you can have kids, you can have your own commitments that that you have for your own
Yeah.
self that were well established that you don't
Yep.
want to break.
Yep.
That's really important for employee retention, I think, long term.
Yeah, for sure. For sure.
Absolutely. Well, we have a tradition here at BioTalk Unzipped in which one of the former guests will write a question to the next guest, or guests in this case, who they don't know who it's going to be. And so they just ask, and we're going to ask you to do the same thing after the show to give us a question that you would like to know something about. So this one is more of a general question rather than a specific scientific question, but how do you measure success in your life? And I'll start with Ines.
Of course, I have to go first. How do I measure success? I think if I'm happy, that's success for me.
That's a good one.
I mean, I don't want to be short, but I feel like, my ultimate goal is to be happy regardless what I'm doing, how much money I'm making. And if I can make the world a better place in the meantime, then that will be the cherry on the top.
100%. Yeah, I think some of the best answers to this type of question are the shortest ones because it gets right to the heart. Yeah. Julie, how about you? How do you measure success in your life?
Am I learning, and are people around me learning from me?
I like
That's
that. Yeah.
that's it. Like if I'm not learning, I'm probably not happy. But sometimes when you're learning, you're unhappy because it's hard to learn new things. So for me, I want to success is I never stop learning, and I'm never stopping trying to translate that and give it to other people.
Excellent. That's great. So, what is the best way for people to connect with the both of you?
Yeah. So if, like we mentioned before, if you sign up for AAPS and join the AAPS WIPS community, we're also on LinkedIn. Like Julia mentioned, we're very active on LinkedIn. So another good way to reach out to us. And please do. And, hopefully this subject touches people's hearts, and we can start being good allies to everyone. It's a both-way street. It's not just, we're not trying to be all about women. It's definitely, we need men, and we need to support each other. Not only men supporting us, but we supporting men as well. Just want to see more of that in our industry. Yeah.
Absolutely.
And the yearly, if you are an AAPS member at FarmSci, just, that's a packed schedule. We realize there's many things that one can see, but we do have a breakfast the second morning that's sponsored by WIPS, and that's another way to find us in real life. And, we'll try to be more communicative this year. I'll personally try to make sure that, we're communicating what sessions we're attending during the FarmSci 360.
Yeah, and we're going to have, meetings. We're going to have a Women in AI meeting. So that should be interesting. We're going to focus on some career development tips. So any women at any level, or men, again, we would like to see more men join the Women in Pharmaceutical side.
We want more men.
Yes,
coming.
we do. Yeah. So please do join the community.
It's not exclusive.
Thank you. Yeah.
Yes.
Yeah.
We
No,
expect
you
two to be joining.
That's what
if you're
All right.
we're gonna
members.
All right.
Be there.
By the way.
The bell's on.
It's our new recruitment mechanism. We go on podcasts and be like, "You'll be joining us, right?"
Join people's podcasts and drag them along. Well, it's a journey. I'd be honored to be dragged along to it. I was going to say, we're honored that you guys reached out to us and said, "Hey, this is an important topic. Would you guys consider bringing it to your to our forum?" And I absolutely honored, and it's been a pleasure to talk to you about it and to really hear, and like I say, learn what we can do and how we can be better, how we can all be better. So thank you.
Yeah.
100%.
We appreciate.
I'll make sure there's links in the show notes down below for both your LinkedIn profiles, and a link. If you can help me with a link to the whips on the AAPS, that'd be great, just to help faster path to that. And I'll also mention that when we get this posted out. So thanks again so much for your time and for coming to us with this great topic. And it's been a real pleasure. Thank you.
Thank you for having us. Really appreciate it.
Yeah, look forward to listening to you guys.
All right.
We should end with the International Women's Day pose, which is take action this year.
Is that it? Is it a flex?
Okay.
Or is it just one? All right.
Or both.
Flex.
Nice.
One for men and one for women?
Yes.
Women and men.
There you go.
Everybody
Fantastic.
can do one.
We made you guys do new, so we can do the flex, right?
Yeah, exactly.
All right.
Awesome.
Thank you.
Thanks so
Good deal.
much.
Thank you.
Thank you guys. Have a great weekend.
Okay. Bye-bye.
You too. Thanks
Bye.
for your time. Take care.